Okay, so remember we are a week out from our next exam. I know it seems like we're still getting over the last one. So that Tuesday before Thanksgiving, that's our exam 4. Some of you have talked to me already. I'm giving you some of you the exam Friday afternoon or Monday morning. So if that's a problem for you, if you're going away, talk to me soon to try to schedule a time to redo that. I really don't want to give it to anybody after Thanksgiving. There could be some situation where that comes up, but I'd rather not have to do that. So let's get it in ahead of time. If you foresee something coming up, somebody will probably be sick or whatever, but let's do that ahead of time. The homework that's due will be chapter 15, innate immunity, chapter 16, adaptive immunity. That's homework that's gonna take you a little bit longer, so plan on that. I want you to take your time doing that homework, look at those animations, think about that. You have to see this stuff a few times before it really sinks in. Also, there'll be a short assignment for chapter 17. So today we're going to wrap up adaptive immunity. I'm hoping that we get a good ways through the vaccine stuff in chapter 17. I also want to include the staphylococcus information on the exam. So we'll either start that today or finish that up on Thursday. I'd like to have a little bit on the exam that's not just immunology. So that's at least what I'm thinking right now, but I'll put out an announcement to be more specific once I know for sure how far we're going to get. Okay, so when we ended up last time, we were talking about MHC Class 1 and Class 2. So I want to just review that a little bit because that's really important to understand that as we get to today, how B cells and T cells get activated and what they do once they're activated. Okay, so do we have any questions before we get into it? All right, so I want to start with a couple review. All nucleated cells in the body. So remember, all of your cells have nuclei except red blood cells, as far as I know. They have the ability to advertise endogenous antigen on the surface of their cells. And they're going to do that by MHC Class 1. So remember, I think one hand. What do you do when you're raising your one hand? You're volunteering. So you're just showing what's on your cells. We do that all the time. And most of the time, our immune system is going to ignore that antigen that we display, as long as the cell's not infected with, let's say, a virus. Okay, so remember this graphic that I showed you. Every nucleated cell has these MHC Class 1. So, one span of that cell membrane. In addition, cells that are antigen presenting cells, like macrophages, like dendritic cells, B cells to some extent, we'll talk about that today. Those also have MHC Class 2 in addition to MHC Class 1. Okay. Okay, so one more question that's important there. So what kind of cells do the MHC Class 1, who are they presenting their antigen to? Who are they hoping is going to notice them? If that is some virus antigen that's been sampled and advertised, Okay, we are hoping that the cytotoxic T cells are the ones that are going to recognize that MHC class 1. Remember, cytotoxic T cells are CD8 positive, and it's that CD8 receptor that actually recognizes the MHC class 1. All right, so we're going to talk more about that today. I forgot to mention, I apologize, I put this on an announcement, but I believe that I posted the wrong thing on Friday. I meant to just post the staff, and instead I posted like 2 or 3 weeks worth of lectures. So nobody said anything like, wow, this is a lot of stuff. But I meant to just post the staff, and I've got that changed now. So ask me if you have questions. Okay, so it's time to kind of put everything together. So we're going to review a little bit about the MHC class 1, a little bit more, the role of different cells. Once T cells get activated, we're going to talk about what they actually do. But their job is to kill cells that are infected with a virus or some other intracellular pathogen. Some bacteria can be intracellular as well. Okay, so I've got 2 different pictures to show you. This one I just got off the internet. It's just a little bit simpler than the one that's in your book. So I always like to see things drawn maybe a couple different ways. It's you really want to kind of look at, see if you understand, you know, it's easy for our eyes to just kind of gloss over a graphic. Really look at it, think about if you had to label it, would you be able to do that? Okay, so let's start out right down here, bottom left. So say one of our cells is infected. All right, so the little red polygons in there, those represent some virus that's in our cell. Okay, we have that mechanism to, just what we just talked about, kind of express a sampling of what's going on in our cell. So the MHC class 1 will present some of that viral antigen. It's the cytotoxic T cells then that recognize that. And our next slide gets a little bit more close up of that immunological synapse. So we'll get to that. But that actually is not enough. We only have a few T cells maybe that might recognize that particular antigen. So you would think once that synapse gets made, and that's going to happen, that could happen really anywhere in the body. You would think, oh, now the T cell is going to be activated and it's going to kill that cell that's infected. But that's actually not enough. We have to get more signals than that because we want to make sure that this is an appropriate response. Again, because it's actually killing our cells. So we have to make sure that that's an appropriate thing to do. So it actually needs other stimulation from a helper T cell. So how does that happen? Well, say some phagocytic cell like a macrophage ingests a virus when it's outside of the cells, okay, and then presents some of that viral antigen here by MHC class 2. Okay, it's the helper T cells that have the CD4 that are going to recognize the MHC class 2 and the T cell receptors that would recognize whatever that epitope. And once that recognition is made, then cytokines are made by the helper T cell that are going to stimulate those cytotoxic T cells to proliferate, make more of themselves, and then to actually carry out that killing. We'll talk about the mechanism that it does that. Okay, so the graphic from your book just goes into slightly more detail. And it draws it a little bit different. It kind of, instead of showing that happening in 2 different cells, it actually, well, it says, well, this could actually all take place within one antigen presenting cell. Okay, so if we start right here, so here's an inactive cytotoxic T cell that's recognizing, say the dendritic cell here, that's what DC stands for, is infected with a virus, okay? And it's got MHC class 1, it's showing what it's infected with. Okay, so over here, is a close-up of that immunological synapse. All right, the dendritic cell presenting by MHC class 1, some epitope, some viral epitope. And remember, an epitope is the part that's recognized of an antigen. And the CD8 that the cytotoxic T cells have recognize the MHC class 1, the T cell receptor recognizes the antigen. So again, it seems like, wow, all those have to be just right. Now the T cell's ready to go. No, it needs additional stimulation from a T helper cell. Okay, so over here, maybe in the same dendritic cell that's also phagocytized some of that virus and expressed it by MHC class 2. The T helper cell with that T cell receptors that recognize that epitope is going to make that synapse, and then cytokines are released from the dendritic cell. And don't worry about which cytokine. Here it says interleukin 12. Don't worry about what cytokine that is. Those are released that cause the T helper cell to differentiate into a T helper 1. There's actually 2 different subsets. T helper 1 cells are going to activate cytotoxic T cells, and as we'll see in a minute, T helper 2 cells activate B cells. Okay, so the T helper 1 cell then secretes, again, cytokines. You don't need to worry about which one, to act on that cytotoxic T cell. Now it is activated, and now it's going to what we say undergo clonal expansion. That means it's going to make more of itself. It's going to copy itself. So make lots more of itself, as well as some memory T cells. So what's special about this cytotoxic T cell is that it doesn't, next time it encounters that antigen, it doesn't have to have stimulation from T helper cells. It's primed and ready to go. Okay, so the next time it encounters that antigen, there's a much shorter response time. But these cytotoxic T cells over here that now are activated, I always think it's funny the way that's drawn. It almost looks like it's a little smoking a cigarette or something there. But that is supposed to be representing the receptor for the particular cytokine that it's secreting to help stimulate it and to keep it active. All right, so we've got a whole repertoire of activated cytotoxic T cells with the specificity for that particular epitope of whatever pathogen. Okay. What happens then once the cytotoxic T cell is activated? I like this figure in your book. It's just a little bit bigger. You can just see it a little bit better, and I left it unlabeled. Okay, so again, I think it's helpful to label these things yourself. Down here, this is the virally infected cell. And here's the activated cytotoxic T cell. Can't see that very well. Okay. Again, label the parts here. This is that CD8 that's recognizing, this is the MHC class 1. Here's the epitope, and the orange is the T cell receptor. All right, so once you have an activated cytotoxic T cell that's recognizing the epitope, okay, then it can bring about the death of that virally infected cell. And there's actually 2 different ways that it can bring about the death. Your book talks about both of them. We're only going to talk about one, and that's the perforin granzyme cytotoxic pathway. Okay, again, this is the mechanism that we think that the cytotoxic T cell accomplishes the killing of that virally infected host cell. All right, and it's really just a couple of cytokines that the cytotoxic T cell is going to secrete. Again, okay, so 2 cytokines, perforin and granzyme. Perforin does what it sounds like perforate. Okay, it's going to poke holes in the infected cells' membranes. Remember, our cells don't have any bulky cell wall to worry about poking through. just cell membranes. Okay, so we see that being released here. It's making a pore, you could say, in the infected cell. And then granzyme is another cytokine that brings about a programmed cell death. And your book doesn't get into any more detail about how that happens exactly. Again, we could take all day on that if we wanted to, but it causes apoptosis, sometimes we call that. It just causes that infected cell to basically undergo a programmed cell death. All right, so unfortunately, that cell can't be saved without, we can't get rid of the viruses in there and still save the cell. It has to be sacrificed, basically. But hopefully it's sacrificed in a way that won't cause all the viruses in there to be able to, lyse out and infect other cells. All right. All right, so moving on to B cells. Okay, remember what B cells do when they're activated. They differentiate into plasma cells and make antibodies. All right, so we talked about that already. There's actually two different ways that B cells can become activated. One way that's a little bit simpler, so we'll talk about that first, is by certain antigens that are considered T cell independent. So that means that there are certain pathogens with proteins on there, typically on the outside of their cell, that those antigens on those pathogens will cause our B cells to just basically activate very quickly. They don't need help from T helper cells. Okay, so a certain kind of antigen, and these aren't very common. It's more common that the B cell does need help from T helper cells. But we call them T cell independent antigens because, again, they stimulate the B cells directly to differentiate into plasma cells and make antibodies. All right, so again, the wording sometimes throws people. T cell independent or T cell dependence. Just got to think through what that means. The T cell independent antigens are often larger antigens, and we said molecules that are large usually are good antigens, elicit a good immune response, but they have lots of repeats. So like a polysaccharide that's, one molecule over and over and over, a monomer with lots, over and over and over again. This is a really fast response. So, when the T cells, let me back up a second here. When these T helper cells get activated, those macrophages or dendritic cells are coming in contact with that antigen. They're typically traveling through the lymph and then have to find, usually in a lymph node, the correct T cell to match up with. So it takes a while for all those matches to get made. If the antigen can stimulate the B cell directly, it's usually pretty, we get the production of antibodies a lot quicker. The problem here, in terms of thinking about trying to get a good response, it's actually overall a little weaker response. In terms of, we only get one kind of antibody made. In other words, there's no class switching. We talked about normally the first antibody produced by activated B cells is, do you remember what kind of antibody? Which isotype? IgM. I think I heard somebody say M. Okay, so normally we get IgM, and then as the course of the infection goes on, the B cells will get signals to change to a kind of antibody that might be better suited to fight that infection. So maybe it's on the mucous membranes and it changes to IgA, or most often it's going to change into IgG because that's the most prevalent antibody in our body. So when you have stimulation of B cells by a T independent antigen, you just get IgM. and it never switches into anything that might be better suited. There's no what we call maturation of antibody. We call that we're going to call it affinity maturation. As an infection progresses, we get little point mutations on the variable region of those antibodies, so they tend to, over time, learn how to fit just a little bit better into those antibodies. That doesn't really happen with these sorts of antigens. And more importantly, there is no memory produced. So, I mean, it is, you know, typically the advantage of having memory cells is that it's able to activate right away. And these do activate pretty quickly, but you're going to have, again, just sort of the same response every time. And another big problem is our immune system doesn't really recognize these antigens very well when we're young. Okay, so this kind of response typically is not generated until about age 4. So some pathogens that have T cell independent antigens, we don't, you know, young children just don't have a very good immune response to them, just because, you know, we say immature B cells, basically. So for instance, here's a pathogen, Haemophilus influenzae, that the capsule that it makes is a B independent antigen. The name makes it sound like it causes influenza, and we named it that because originally we thought this bacteria did cause influenza. It turns out it doesn't cause influenza, but it does cause meningitis. All right, and so anytime a young child gets bacterial meningitis, it's a very serious thing. So young children are especially vulnerable to this reaction. As adults, we fight this off really well because we have a good response to these type of antigens. But this is why it's so important to immunize young children against Haemophila. It's a Haemophilus influenzae. It's that Hib vaccine. So Haemophilus influenza B. Okay, so again, sometimes, you know, adults, why do I have to give this to my baby? I've never heard of this, and, you know, I'm not going to get sick with this myself. But yes, young children can. Okay, so this is the graphic that used to be in your book, and the later versions cut it out. It's always good to have a picture to look at. So here's an example, some polysaccharide with repeating subunits. Not all polysaccharides, just certain ones, it seems like, often function as T independent antigens. Here's a B cell receptors recognizing that. And again, there's no T cell help here needed. The B cells will differentiate into plasma cells, and the plasma cells are making IgM. They didn't draw those as a pentamer like we usually see IgMs, but okay, typically IgM. And again, over here, we've got no memory cells. Okay, so not quite as good a response, but a very fast one. More often, antigens are T cell dependent. So in other words, to elicit that B cell activation, we need some help from T helper cells. All right, and again, it takes time for those immunological synapses to kind of find each other and take place. That's often have to travel through the lymph and find each other in the lymph nodes. But let's walk through how that happens. So let's start right here. So here's a close-up. You've got a antigen presenting cell, again, like a dendritic cell or a macrophage, that has phagocytized some pathogen. So this is going to be some extracellular pathogen, say a bacterial cell, and presented it by MHC Class 2. Okay, and it's the T helper cells. It's kind of hard to read that up there. T helper cells with the CD4 that recognizes the MHC class 2. Don't worry about these other like CD828 or CD60. Don't worry about those. Okay, once this synapse has been made, now the T helper cells are going to get the signals to differentiate into T helper 2 cells. Okay, again, don't worry about what cytokines are important there. At the same time, over here, we've got some B cells that have ingested the same antigen and presented it by MHC class 2. So B cells are not really phagocytic cells, but they do have a mechanism to kind of ingest and present antigen by MHC class 2. Okay, so they're presenting that antigen, and now we have the B cell finding those activated T helper 2 cells, okay, probably in a lymph node somewhere. And once that connection is made, the proper cytokine signals will cause the B cell to activate, all right, and make clones of itself and clone and make plasma cells. And again, those plasma cells create the antibodies, as well as we get some memory cells. Okay, so these are B cells that are activated that can persist for a long time in our body. And once those B cell receptors see that same antigen again, they don't need the signals from the T helper cell anymore. They can just start making antibodies themselves. Okay, there is an animation as part of your homework. Yeah, question. Yes, so question is, can I go over how the B cells get the antigen and present it again? So you have a repertoire of B cells, and B cells can actually ingest antigen that's not quite like phagocytosis. It's kind of similar to that, but they can ingest antigen in kind of a membrane-bound vesicle and then present it on their surface with MHC class 2. Okay, so they have kind of special mechanism to do that. And then when those cells, probably traveling in the lymph, are going to meet up with the T helper 2 cells that have been activated in the lymph, okay, then cytokines are released that will activate the B cell. Okay, so it's a lot of kind of, you know, things that have to be met. Everything has to work out just right in order to activate those cells, because we don't want them activated unless it's appropriate. Okay, we don't want to activate against things that don't require the production of antibodies. Okay, it's a pretty good animation of that as part of your homework. So humoral immunity, remember, humoral immunity just involves antibodies. Humors are your bodily fluids, so we typically, that's what we call kind of our antibody response. Clonal selection, that means like which B cells with which B cell receptors are going to be cloned, made more of them, and expansion. Okay, so select which B cells to make more of. All right, so that's a good animation. I definitely, spend some time looking at that. All right, so here's the so what. So if we look at a primary and a secondary response to some antigen, a primary response meaning the first time our body sees that antigen, and the secondary response meaning the second time we're exposed to that antigen. Okay, so what happens a little differently? So we'll use the example here. Say a person is given a tetanus immunization. For tetanus, we have a special vaccine that we call a toxoid vaccine, because for that infection, tetanus, it's the toxins that cause all the damage. So we actually give an inactivated tetanus toxin as a vaccine. So say a person gets a tetanus vaccine here at time 0. What happens in their body when they're exposed to that antigen? It's a little bit of a lag period while all those the cells are finding each other and get the appropriate, T helper cells get activated until the first antibody is produced, IgM. Typically, after a while, that might switch to another class, usually IgG. And here we're out day 15, so 2 weeks after. So it takes a little time to get those specific antibodies made. But then the next time that host is exposed to, say, maybe they're exposed to the real thing. Maybe they step on a rusty nail and there's some Clostridium tetani in there and it starts multiplying in the body and producing tetanus toxin. Okay, so that's time 0. What happens in the body? Well, you've got some memory cells from the first time, hopefully, some memory B cells. And so we get a faster response. Here we're getting really the beginning of antibodies produced maybe within a day or so, rather than maybe day 3 or 4 as the first time. And we're right away getting antibodies that will best fight that particular antigen. So instead of just IgM, we're also class switching right away, having plasma cells that make IgG, and in much higher amounts. Okay, so we get more antibody. We would say a higher titer or higher concentration of antibody more quickly. Okay, and it's all because you've got these memory B cells and T cells, and T helper cells, memory T helper cells that require less stimulation to activate. And if there was a tertiary response, hopefully it would be even a little bit quicker than that. Okay, so that's one of the hallmarks of adaptive immunity. We learn over time and get quicker response. All right, so these are terms that I mentioned before: isotype switching, the change of antibody class. So the B cells and plasma cells get cytokine signals when it's appropriate to do that. And over time, a little affinity maturation. So subtle point mutation changes in the binding of the antibodies that are produced in order to, again, better fight the infection. All right, so this is all with T-dependent antigens. So backing up just for a minute, what about T-dependent antigens? All right, so remember, those are the ones that don't require T helper cell help. What would the primary and secondary response look like with T cell independent antigens? All right, so here's the graph of T-dependent antigens. It's just drawn a little bit different way than the one that I just showed you in the book, where the second time we get more class switching, more antibody. What if we drew that same graph for a T-independent antigen? It should look something like this. And I didn't post this slide. Remember, we got no memory cells. So same thing. Second time you come in contact with that antigen, same thing. Only IgM antibodies, same amount, because no memory from the first time. Okay. All right, do we have questions? Anything you want to clarify? Yeah. So explanation of IgM and IgG. So those, remember, we have 5 different isotypes of antibodies. The M or the G or the A or the E, that all refers to the stem of the antibody. So remember, the antibodies look something like this. The stem, this is the FC portion. It's either going to be, they come from a certain gene segment, basically. So we name them based on the stem region. The variable region is up here at the top that binds to the epitopes of the antibody. So we talked about how different kinds of antibodies are kind of better suited to fight different sorts of things. The IgA is often the one that's present in mucous membranes. It's found in our bodily fluids like tears and breast milk. IgG is the one that crosses the placenta and is a little bit longer acting, so it protects the fetus even into their first few months after birth. The IgE, remember what the IgE does, what it's good at? It's very prevalent whenever we have an allergic reaction, but it also fights especially parasitic worms. We see a elevation of IgE antibodies in our bloodstream when we're fighting some, helminth worm infection, which doesn't happen very often. Okay, IgD, we don't know a lot about that one, so we don't talk much about that. All right, does that help answer your question, Megan? Okay, another question. No, you don't need to recognize the stems. Like, to me, they all kind of look alike. We did say that, like, the IgM is the one that's usually as a pentamer, so 5 of them together. The IgA often exists as a dimer, so those are a little bit more recognizable. But the G and the D and the E, I wouldn't be able to tell those apart. Yeah, question.
So going back to the antibody production by T cells, so you said antigens are presenting cell phagocytizes a pathogen and presents it by MHC Class 2, and then you said something about CD4 cells.
Right. Okay. So let me just back up to. I think what you mean is. Whoops. Here. Okay, is this where you're asking? Yeah, so up here, the T helper cells, remember T helper cells are CD4 positive, cytotoxic T cells have that CD8 positive. Okay, so the CD8 recognizes the MHC class 1, the CD4, you can see here, it's this pink right here recognizes the MHC class 2. Yes, yeah. Okay, good. Another couple. Megan next, and then it stands for cluster of differentiation. They're considered glycoproteins that are on the surface of that particular kind of cell. It's like a receptor protein, basically. Yeah, yeah.
So going back to the slide that's killing by activated T, like cytotoxic T cells, so isn't that
So going back to this T cells, you said, so like this slide right here. Yeah, so I'm assuming that it's already activated. I was just kind of giving you a little practice labeling that again, and just assuming that it's activated. What's it actually going to do? Then it's going to do this, bring about the killing by the perforin granzyme. Does that answer your question? Yeah. Okay. Okay. Good. Don't be afraid to ask. Clarify. Okay. So to finish up our adaptive immunity, here's a really nice segue into our next section talking about vaccines. So how do we acquire immunity from some pathogen? Okay, there's really 4 ways that we can describe that can happen. And this is, really good to think about, especially as we are living through a pandemic. Okay, so we ideally want to have acquired immunity from COVID, right? So what are the 4 ways that we can get that? So the terminology here is pretty easy once you kind of get it sorted out. We have 4 different ways to describe how we can get that immunity, either naturally or artificially. Okay, so if we get it naturally, think about, could we have gotten this 500 years ago? Or does it require some bit of technology or some, human intervention? Then it's going to be artificial. Okay, so I'll give you some examples of both. And then we describe it, immunity, as either active or passive. If it's active immunity, it means we make antibodies ourselves. And if it's passive, that means antibodies are just given, just kind of passed on, I guess you could say. Let's see, how could I say that better? All right, yeah, so active patients, the patients making their own antibodies, and passive immunity is they're getting preformed antibodies from someone else. Okay, we'll say from someone else. Okay, so I think this works better to give some examples. So if you become immune to a disease due to naturally acquired active immunity, that means that you have contracted the disease, and as a result of that, you have memory B cells and T cells, and so you're not going to get the infection again. There's lots of diseases where you get it, or there's some diseases where you get it once and then you typically never get it again, because you have, again, those memory cells that basically, you make the antibodies and don't even know that you get exposed to it again sometimes. Okay, so normal life experience, exposure to pathogens. I got chickenpox when I was 8, and now I have good active immunity against that. I can make, antibodies if I'm exposed to that again. Natural passive immunity then is when you get antibodies from someone else. So, and the only way that can happen naturally is through, or the way I can think of, is through breast milk. Okay, so a mother passes that to their child. Again, did they do that 500 years ago? Yes. Okay, so we'll call that natural. It's a short-lived protection, so only as long as those antibodies are circulating in the newborn. It might be, a month or two, but they have immediate protection from that pathogen. Artificial active immunity, that's when we get a vaccination, right? So like that tetanus toxoid, we were exposed to that. We make memory cells from that, and then the next time that we're exposed to the real thing, we have a good defense against that. And then artificial passive immunity is when we get antibodies by some infusion. Okay, so they did this a bunch with Ebola. They harvested antibodies from people that had survived Ebola and gave it, through IV to people that were sick with Ebola to try to give them another weapon to fight that. They are doing this with COVID now, especially if somebody has some risk factors and you find, and you catch it kind of early on in the infection, they will give you an infusion of antibodies. So this is artificial because, right, they didn't do this 500 years ago, right? They didn't have syringes, they didn't have the technology, all right? So we consider that artificial. Another name for that is immune therapy. All right, so again, the key is there to, natural, know the difference between natural and artificial, and active and passive, and then you can put those words together. Okay, so again, that dovetails really nicely into a little discussion here of vaccines. And so we're getting to chapter 17 here. We're only doing just a little portion of chapter 17. It talks about in the book some kinds of immune testing, ELISAs and stuff that we're not going to get to. This used to be a really short section of my presentation because I only talked about vaccines that were approved for use in the US, and that list has gotten a little bit longer. So I have a lot that I want to say, and hopefully I'll say it coherently, I guess. It's a very interesting topic to me. Vaccines, there's always been some resistance to vaccines. Okay, so I found this picture online. It was put out by the Anti-Vaccination Society in 1802. All right, so our first vaccination was around 1798. Edward Jenner realized that milkmaids that got cowpox very rarely ever got smallpox. So he took those cowpox scabs from a milkmaid and inoculated a kid, like with a knife, poked those scabs into his arm. And then a few weeks later, he inoculated that kid with smallpox. Not the way we do clinical trials today, but the kid lived, and that became a very common practice, very non-hygienic, like, you know, people probably got staph infections all the time from doing it that way. But people kind of made fun of and resistant to things that they didn't understand. So, you know, I don't think they really thought that, you know, women were throwing up cows or giving birth to cows, or, you know, I don't really think they thought that. But again, they were resistant to the idea of this because we didn't really understand how it worked. And we still see that today to a big extent. All right, so could not resist the Aaron Rodgers reference here. This was on my slide already. What's the difference between vaccination and immunization? Most of us consider those things the same thing. Credit to Aaron Rodgers, though, he knew there was slight difference between those two terms. So what is vaccination is basically when we get a vaccine. Okay. Immunization technically is what happens in your body when we get vaccinated. Okay, so semantics there, slight difference. You know, usually when we talk about getting immunizations, we assume, oh, you got a vaccine and then you were immunized. All right, so you got memory cell production. In his mind, he took some kind of holistic medicine that he's not really telling us what that was, to beef up his immune system, not to any specific kind of pathogen. So, you know, it was kind of misleading when he said, oh, yes, I've been immunized. Maybe in his mind he was, but probably not really. All right, so why do we want to vaccinate? Not just to be controlled by the government, right? But we want to vaccinate our vulnerable populations of people. So we want to especially protect children. There are some pathogens that, because they have an immature immune system, they're not going to have a good response to, especially those T-cell independent antigens that we just talked about. Haemophilus influenzae is an example of that. We also want to protect older adults because as we age, our immunity wanes. We don't have as good a response as we get older. So there's a number of vaccines that are recommended when you turn 50 and when you turn 60 or whatever. So, you know, talk to your parents and grandparents about what vaccinations they should be getting. The whole point is to try to achieve herd immunity. And we hear that term thrown around quite a bit these days. Basically, that refers to how many people in a population need to be immune to something until that pathogen dies off. Okay, so there's a lot of discussion about how many people need to be immune until, you know, COVID just goes away. We don't really know. For a lot of infections, it's somewhere between 70 and 90% of the people. Okay, so the thinking is if only, you know, certain people are susceptible, those people are not likely to come in contact with each other and the disease will die off. We know that for measles, herd immunity needs to be like 95% because measles is so infectious. That Delta variant of COVID is extremely infectious as well, probably as infectious as chickenpox. And, you know, that's an illness that just everybody got. So, the question is, can we even get to herd immunity? The idea is that we protect, if we can get to that herd immunity, then we can protect people that can't be vaccinated because of maybe, some pre-existing condition or lack of immune function. Yeah, I don't know. I don't know if we'll get there. Maybe there's some promising drugs that we'll talk about when we talk about COVID. So maybe that will help us get there. Okay, the goal of vaccination here, again, is to get good memory cells. Hopefully both memory B cells and memory T cells. Okay, so we want to induce adaptive immunity. Whoops. Okay, in order to get memory cells. Ideally, we're going to get both T cells and B cells. Sometimes B cells are enough. Okay, it's a little bit harder to get the memory T cells. All right, so let's look at the different types of vaccines that are approved for use in the US. Okay, again, this used to be a shorter list than what it is now. Our very first vaccines that we had were attenuated vaccines. And that just means a weakened strain. Okay, so for instance, that smallpox vaccine was really a milder infection, cowpox. And it was similar enough to smallpox that it would keep somebody from getting smallpox. And that was kind of a risky way that they used to inoculate people with that, but smallpox has a mortality rate of about 30%. So, you're always just weighing risks, like, oh, what are the chances I'm going to get some kind of secondary infection around that site? Or vaccines are a lot safer now than what we used to do. So in the attenuated vaccines, that pathogen has been modified in some way so that it can no longer cause disease. And that's done a lot of different ways. Sometimes we grow it in a cell type, like for a viral pathogen, we grow it in a cell type that's not its normal host. So for example, the polio vaccine, the Sabin polio vaccine, we grew that in monkey kidney cells rather than human nerve cells. Okay, and that caused enough mutations that then it wasn't able to cause an infection in us. Sometimes we grow it at a different temperature or just pass it for many, many generations, grow it up for a long period of time. That's the tuberculosis vaccine that we have. That's what we've done. And we don't really understand the mutations that have taken place, but we know that it's, or we feel that it's pretty safe. The advantages of attenuated vaccines is that they are going to enter our cells very naturally. And so we get a really good, presentation by both MHC Class 1 and MHC Class 2. We get a more natural adaptive response. So we usually get really good memory cells. It usually only requires a dose or two in order to give good protection. One thing that can be considered an advantage, but we actually usually call it more of a disadvantage, is that it can cause a mild form of disease and therefore spread contact immunity. So when might that be an advantage? If you're trying to vaccinate maybe a remote population of people, maybe, in the rainforest or jungles of the Amazon or something like that, and you can only get there once. Well, if you give it to a small number of people and it causes mild disease, and other people that didn't get vaccinated could catch that mild disease from other people, then you could spread the immunity that way. Disadvantages of attenuated vaccines, that contact immunity in the US anyway would be considered a disadvantage. Possible to have transmission. We really don't want to pass it on to others, because what if that other person is a baby that maybe, you know, has an immature immune system, or we pass it on to somebody that's immunocompromised, and then it's not causing a mild disease, maybe it's causing a severe disease. So generally, we don't want to, you know, one vaccinated person don't want to pass it on to somebody else, transmit it. Usually you have to refrigerate, because again, these are live strains. Either it's a virus in a live cell, or, you know, a bacteria that's, again, alive. So you got to refrigerate it, more difficult to take to remote populations. And really the biggest reason here is the small chance that you would have a reversion back to a wild type, meaning the normal pathogen. An example of when that happens. So we actually have we had two polio vaccines, like polio, huge scourge in the 1950s. Two polio vaccines were developed. One was an attenuated vaccine, that was the Sabin vaccine. And one was actually just part of the virus, a subunit, we'll get to that next. And that was the Salk vaccine. And so we kind of went back and forth as to what version we used. They decided Sabin vaccine was better. That was the attenuated version. And that one also had the advantage that it was oral, meaning that you would just give it to kids to drink, one less poke, you know, so that was kind of a nice thing. Well, in 2000, they were giving the Sabin vaccine to some kids in Haiti, and about 20 kids got polio from the vaccine. All right, so somehow that weakened strain of polio, there was some mutation that happened and it reverted back to the polio that causes full-blown polio. So after that disaster, basically, we never used that Sabin vaccine anymore, and we switched back to using the Salk, which doesn't work quite as well, but at least there's absolutely no chance that somebody's going to actually get polio from it. So typically, we don't use an attenuated vaccine unless we don't have another one that works. So some examples: measles, mumps, and rubella MMR vaccine, yellow fever. We have a lot of different kinds of influenza vaccine, but the one what we often give to kids of a certain age, it's a strain of influenza that can only multiply it when it's a little bit, the temperature's a little bit lower. So like in the nose, nasopharynx area, it can't multiply once it gets into the lungs. So it causes a really mild infection and works better, I guess, for kids especially. All right, so also chickenpox is an attenuated vaccine. Okay, so again, usually only have to take, you know, 1 or 2 doses of it, but there are some risks involved there. More commonly, our vaccines are killed either whole cell or subunit. So we'll give examples of both there. Sometimes we call these inactivated vaccines because if it's for a virus, well, was a virus alive to begin with? We don't really consider them alive, so sometimes we call them inactivated. All right, so the nice thing is these are, because they're not alive, they're not going to replicate in the host at all. We do need some more boosters in order to get a good level of protection. There's just generally less antigens, less epitopes. It doesn't enter the cell quite the same way as a live virus, say, for example. So we mostly get a humoral response. Remember, that's a B cell response, because it's difficult to get presentation by MHC class 1 if it's dead. Like our cells just don't, again, take them up or phagocytize them the same way. So killed vaccines, most of the influenza that adults get anyway, rabies, Salk, polio. The old commercials for U of M, they used to show a picture of Jonas Salk. They like to take credit for his work. He actually did some of his work at U of M, but then he went on to University of Pittsburgh to really develop the vaccine pertussis as well, also known as whooping cough. All right, so a kill vaccine is either the whole cell that's been killed, or it's just part of it. Okay, so a subunit vaccine, we also would call that acellular. So it's maybe just some protein, just some vital parts that causes has good immunogenicity, good antigenicity. All right, so if it's a whole cell vaccine, you treat it in some way in order to kill the cell. So either with heat or by some chemical means, formalin, maybe even radiation, hit it with gamma rays or UV rays, maybe, I'm not sure. Inflammation here can be a problem. If you think about, it's a certain case, if you have a gram-negative whole cell, what do they have in their outer membrane that can cause inflammation? They have endotoxin. Remember, that's that lipid A part of the lipopolysaccharide. When the cell dies, the cell wall starts to break up a little bit, but some of it will still be intact if it's a whole cell vaccine. So we get some endotoxin delivered with it, and that can cause a little bit more inflammation. So the pertussis vaccine used to be whole cell. So it's given along, the D is diphtheria, the T is tetanus, and then the P is pertussis. Okay, so this was the whole cell pertussis. And inflammation, like, some redness, some achiness, maybe a little fever, things that go away after a few days. But when you give your baby that vaccine and they're cranky for a couple days, mothers are less likely to go back and get the next dose. Like, my kid was a handful, I don't want to go through that again. So that's a problem. A subunit vaccine, we switched actually because of that inflammation. Got ahead of myself a little bit here. Now we give pertussis. It's just a portion. I think they take a little bit of a surface protein and maybe a little bit of the pertussis toxin and put it in the vaccine. So now we give diphtheria, tetanus, and AP stands for acellular pertussis. So instead of giving the whole cell, we just give a part of it. It does have just fewer epitopes, basically, for us to react against. So the protection is not quite as good. Typically now, there was kind of a fuss about this before the pandemic, how that pertussis vaccine usually wears off by the time kids are like 8 or 9. They're supposed to get a booster when they're 12, but that's not a time when we really often think about giving our kids boosters. So it's not as good protection, but at least we protect the babies, which is when people are at most risk of having very severe pertussis. Okay, so more boosters, but less problem with inflammation, so people are more likely to vaccinate their kids. All right, and then a third type of vaccine that we've used for a long time are toxoid vaccines. There are a couple infections where the damage that the infection causes is due to the production of a toxin. All right, so we talked about one already, tetanus. That tetanus toxin, it's actually a neurotoxin. We just basically take that toxin and inactivate it, again, sometimes by heat or chemical means, and that produces a pretty good antibody response. You don't get very good cell-mediated response, but that typically is enough. Those antibodies can bind to that toxin and neutralize it. Another toxin vaccine is the diphtheria toxin. And diphtheria is an infection that you'd never hear about anymore. It used to be a huge scourge. This is a patient with diphtheria. You get these membranes in your throat, and especially dangerous in kids, and it basically causes them to asphyxiate. They can't get airflow. So again, severe disease, there's like almost no cases of it in the US anymore, thanks to the vaccine. All right, so moving on to vaccines that are approved. So the messenger RNA vaccines that are approved now, the Pfizer vaccine, the Moderna COVID-19 vaccines. We have been doing research with messenger RNA for over 30 years. At first, that messenger RNA was touted as, we were really hopeful that we could design drugs that were messenger RNA drugs. And there are some reasons that hasn't panned out very much so far, but it's the hope that they may in the future, in addition to having vaccines, perhaps, some cancer treatments as well. So this technology holds a lot of promise. One advantage of using messenger RNA vaccines is that they are very safe in terms of that messenger RNA is not going to cause COVID. It's a piece of RNA that encodes for the spike protein. Okay, so it's just a little piece of that COVID virus. It's not going to cause an infection all by itself. It's not going to mutate into COVID-19, and it's not going to mutate our cells, right? We never make DNA from RNA. In order to produce it, it's very quick and easy to produce. The influenza vaccine, we'll talk about that more a little bit later, but we have to grow that up in eggs, and it takes months and months. So we start producing influenza vaccine in February before, the year that we use it. So it takes a long time to make. The Pfizer vaccine, we had the first messenger RNA like a week after we'd sequenced COVID. And then it took 8 or 9 months to make sure that that was safe. But we produced that right away. Of course, it takes a while to scale it up. But disadvantages. So that messenger RNA degrades pretty quickly. Both when you put it into our bodies, it degrades quickly. And that took a while to kind of monkey with it a little bit, to protect it a little bit, so it didn't degrade from our endonucleases as quick. And also, outside of our bodies, right? We have to store it, minus, I think it's minus 80 degrees Celsius. I think we have to keep it really cold. We have to store it so it doesn't degrade. And as most of us know, short-term inflammatory reactions are pretty common. Things like, redness, achiness, headache, those sorts of things. So why that is, we really don't want an innate reaction. And unfortunately, we have to call in some of our innate immune cells in order to stimulate our adaptive immunity. But that messenger RNA is actually, it's viral, right? And it's a PAMP, it's a pathogen-associated molecular pattern. So when that's injected into our body, our macrophages that would see that, they have the toll-like receptors on the outside, and it's actually TLR7 and 8. So toll-like receptors 7 and 8 are the ones that recognize foreign messenger RNA. So if you think back to our chapter 15, those are expressed once the macrophage chews up the messenger RNA, and it's on the endosome, like inside the macrophage. So once it recognizes that pathogen-associated molecular pattern, whatever it is about viral messenger RNA that looks different than our messenger RNA, that starts to, pulls the burglar alarm, starts to trigger more innate immune reactions, more inflammation. So we've done a number of things to try to avoid that a little bit. We've actually used modified nucleosides. They're supposed to look more like our messenger RNA, even though they still code for that viral spike. So most of the inflammatory reactions, again, are short-lived. There is a more serious one, myocarditis, that's associated with both Pfizer and Moderna. It's rare, 1 out of 50,000 people, a little bit more common than that in men, a little bit less common in women for some reason. And that's characterized, typically patients feel kind of a tightness in their chest. And usually it's an inflammation of the heart muscles. That's what myocarditis is. And usually that goes away after a few days, but that has the potential to be serious. Again, it's pretty rare. And actually myocarditis can be much more commonly than that a symptom of having COVID. So if you get COVID, you're more likely to get myocarditis than by getting the vaccine from the vaccine. So again, it's all about balancing the risks and have the best data that we have. Okay, and that's again more likely after the second shot. I don't know about the third. We don't have any data about that yet. And then really closely related, the Johnson & Johnson vaccine, that's considered a viral vector vaccine. So we talked about what vectors are. Usually a vector is an insect that transfers a disease, from one host to another. In this case, we're using a virus. We're using a modified adenovirus. So we actually had an adenovirus drawn out when we were talking about viruses. That's the picture that we had, the hexon and penton proteins, capsomeres that we talked about. So an adenovirus is we named that, it was isolated from someone's adenoids. So that's where the name came from. Adenoviruses are usually causing really mild, kind of cold-like symptoms. In this case, they've used a chimpanzee adenovirus just so that it couldn't replicate in our bodies. All right, so the adenovirus can't make more copies of itself, so that's pretty safe. It's just a vector. It's a way to transfer that DNA, in this case, into our cells. Okay. So again, it enters the cells, but it can't replicate. DNA is a little bit more stable than RNA, so we don't have to keep it as cold outside. It doesn't degrade as quickly inside our cells either. I should update this. Now we're giving boosters for Johnson & Johnson as well. We think that it works even better with 2 doses. And the way this works, so once you get the DNA inside our cells, right? So the DNA gets made into messenger RNA, the messenger RNA gets read at the ribosomes, and then we get that spike protein expressed on the outside of our cells, right? So that's the whole point, to make that spike protein. Okay, so our body's immune cells recognize that as foreign, and we'll get an immune response to that. In addition, we get some presentation by MHC Class 1 of that same spike protein. Okay, so that's being made in our cell. So this way we get, the cytotoxic T cells involved. The spike protein will get phagocytized. Macrophages then will express that protein by MHC Class 2 that will get the B cells involved. So we get pretty good, B cell and T cell involvement there. Disadvantages with the Johnson & Johnson, again, there's some pretty rare but very serious side effects. There's a clotting disorder that has been, let's see, the numbers that I found here. This is about 1 out of every 500,000 doses. So pretty rare, but that's pretty serious. Guillain-Barré, chances there, 1 out of every 120,000 doses. Guillain-Barré can be, that's like it affects the movement of the muscles. I think it has some neurological implications as well, but I think it's a weakening of the muscles. Typically goes away and gets better, has the potential to be serious. Any viral infection, rarely Guillain-Barré can be a complication of any viral infection. So it can be a complication of COVID as well. Okay, and then I want to talk a little bit while we're talking about COVID. We just got a couple minutes left here. What does it take for a vaccine to be approved by the FDA? So this I just copied out of the CDC website. And before we even get into clinical trials, the vaccine has to be tested in animals. Okay, now we may not have a good animal model for some pathogens. We might not have an animal that shows the same symptoms as what we do, but at least we can test to make sure that the vaccine doesn't harm the animal in any way. Okay, we can figure that out before we get to testing in humans. And then there's several different phases of when we get, okay, let's say it looks like it's safe in animals. We test it. Phase 1 is a very small group of volunteers that are considered healthy. So we vary the dose a little bit maybe, and we look for side effects and try to see if it actually works. Once everything looks okay there, we go to phase 2. It just involves kind of the same kind of thing with a few hundred volunteers. And then in phase 3, again, hundreds of thousands of volunteers. And you have to give at least 2 months after that to look up for any side effects. Okay, we are out of time, so we'll talk about phase 4 next time. And have a good day.